Okay, this is our discussion of the general and special senses, and this is how the body is able to gather information about stimuli that we're either consciously or unconsciously aware of, and then turn that into electrical information, send that information back to the central nervous system, which is either going to be processed by a region in the brain called the cerebral cortex or um, a region in the central nervous system called the hypothalamus in the brainstem. Okay, and then commands will be issued based on that information, designed to enhance our survival. Okay, so we'll talk about what the components of the sensory system are and where they're located. Talk about the five senses, and we'll go into more detail about smell and taste and vision and hearing and balance. Okay, so our sensory system lets us see, hear, and taste things in our daily lives and acts as a warning system against harmful stimuli, and it keeps track of what's happening inside the body and sends signals to different organs and helps maintain homeostasis. Okay? Some of this information we're consciously aware of, and some of this information we're not. Okay? Um, Examples of information that we're consciously aware of: sounds, light, color, smell. Okay, um, senses on the skin. All right. Information that we're usually not aware of would be information, stimuli information from our internal organs. Okay, such as a blood pressure change or a glandular secretion. All right, and that stuff is still dealt with, but we're not consciously bothered by it because it would interfere with our ability to really deal with and interact with the world around us. A receptor is just a specialized cell. It could be a neuron or it could be a sensory receptor, okay, that picks up a particular type of stimulus. Okay, chemoreceptors respond to certain chemicals in the environment. Pain receptors respond to pain, which can come from interaction with chemicals or physical force or temperature, just to name a few things. Okay. Um, Thermoreceptors respond to temperature. Mechanoreceptors respond to physical change. Okay. Twisting, pressure, vibration, so on. Okay. And photoreceptors respond to light. Okay. Where are these found? Well, chemoreceptors are found in our sense of smell, right? So they're in the nasal cavity for one place. They're also in the tongue, okay, where they respond to things that provide taste. Um, Pain receptors are pretty much everywhere, um, with the exception of the central nervous system itself. We have pain receptors throughout the body, and that's to warn us of damaging stimuli so that we withdraw from it or we treat whatever's causing the pain. Thermoreceptors are found in the skin, okay, and also to some extent in your internal organs. And mechanoreceptors are really all over the place. They're in your skin, they're in your joint capsules, they're even in your internal organs. And photoreceptors are found primarily in the retina of your eye. Okay? The four components of sensation, okay, are essentially the stimulus, right? That hits the receptor. The receptor generates a chemical that causes a sensory nerve to generate an electrical impulse, and that gets relayed to a region in the brain or the brainstem. Okay, and then from there, um, that's what we would call sensation. Okay, and then what the brain does with that is it applies meaning to the sensation. So that's what perception is about, and perception draws on other regions of the brain and uses memories from those regions in order for us to understand what's happening to us. Okay? Characteristics of sensation include projection and adaptation. Okay? Projection is where the brain refers a sensation back to its original source. which would be an ordinary injury, or in an unusual case called phantom limb pain, where we're missing a piece of the body, but we still get pain information because of the damaged nerve endings and the brain's map of the body that still exist. Okay? The reason that you see with your eyes, you hear with your ears, and you feel pain with your injured hand is because the cortex of your brain gets the sensation and projects it back to its source. The brain receives the sensation of the painful thumb in this diagram and refers it back to its source. Okay? Adaptation is the loss of sensation with continuous stimulation. Right? The sensory receptors get less responsive, and over time, the sensation appears to vanish. Okay? Receptors are going to vary. Some receptors are going to adapt quickly. Some will adapt slowly, and some won't adapt at all. Okay? Olfactory receptors are quick adapters. Receptors that determine body position and pain adapt slowly, if at all. It's beneficial that we have that pain and body position information all the time. Otherwise, if we didn't have body position information on the fly, we wouldn't be able to stand up straight and interact with our environment. And if we didn't get pain information, we wouldn't treat the area that's affected. Okay? Note that prior to a diagnosis, pain is not medicated so that we can determine the symptoms. Okay? And we should also note that we can have both general and specific types of senses, right? Our specific senses are taste, smell, balance, vision, and hearing. Okay. Our general senses are generally those in the skin and in the walls of our organs. Okay? So those are some general senses, right? Pain, touch, pressure, temperature, proprioception, all right? And there are receptors in the skin that deal with all of that. Okay? And they have nerve endings that are modified to generate an electrical signal when that particular stimulus happens. Okay? So a touch receptor, for instance, would never generate a pain signal, and a pressure receptor would never generate a temperature signal, okay? Because it doesn't have the right modifications to do that. Okay? Pain receptors are also called nociceptors, and they have free nerve endings and are stimulated by any type of stimulus that's outside the normal stimulus range for a thermoreceptor, a chemoreceptor, a mechanoreceptor, and so on. Okay, so the response to stuff that's outside that range is always pain. Okay, so they're where? They're all over the skin. They're in our organs and they're in our tissues, and they don't adapt. Okay. Pain triggers that have been identified include tissue injury, oxygen deficiency, and mechanical force. Okay? An example of how this works is that if you apply pressure to, let's say, your finger, okay, you can apply light pressure to your finger and you can feel that. You can apply heavier pressure and you'll experience that. But if you say slam your finger in the door, okay, you're not going to feel pressure. You're going to feel pain because the pressure is outside the range for the pressure receptors that are in the skin and the joints. Okay? Now, referred pain is a different animal. Okay, with referred pain, what we have is pain that appears to come from a region of the body where the pain isn't actually being produced. Okay, and the reason for this is that the nerve tracts that provide the pain from those regions of the body and from our certain parts of our internal organs travel along the same tracks up to the brain, and the brain has a difficult time distinguishing between the two. Okay, so for instance, the pain of a heart attack, right? You can see here is radiating pain along the neck, the left jaw, the left arm, and through the left pectoral region. And it's actually coming from the center of your chest, right? That's where your heart is located. But your brain can't determine that because the pain leads from those regions we just described, and the pain leads from the heart, travel along the same pathways to the brain. Okay. Note that these impulses are going to travel up the spinal cord, okay, through something called the spinothalamic tract, and then it gets transmitted to the brain's router, which is called the thalamus, which sits in the core of the brain, and then that's going to project that information to the parietal lobe of the brain in what's called the primary somatosensory cortex, which lies like a barrette across the middle of the brain going from ear to ear, okay, and is mapped to different parts of the body in proportion to the number of nerve endings that come from that part of the body, okay. Analgesics can relieve pain by interfering with the transmission of those signals or by interfering with the function of the pain receptors, okay. We can do this also with current, right? TENS units can pass current through the spinal cord and can interfere with ascending pain signals, okay? And that's one of the ways that we treat pain without the use of narcotics, right? Tactile receptors respond to touch, okay? These are mechanoreceptors, and they're found mostly in the skin. And they're modified to respond to that type of stimulus, okay? Pressure receptors are also mechanoreceptors, and they lie in the skin and in the subcutaneous and in the deep tissue, and they have modifications around their nerve endings that open ion gates when the pressure is applied, right? So when you mash down on these nerve endings, little tunnels in the membranes of these nerve endings open up, and then current flows in, and that produces the sensation of pressure. Okay? Thermoreceptors respond to changes in temperature, okay? And these are free nerve endings and specialized sensory cells in the skin that adapt relatively quickly, and if you apply a temperature change outside the range of those receptors, you will experience pain. The nociceptors will fire, okay? Why is perceiving temperature extremes protective? In order to avoid tissue injury, right? So that's why we don't want to put our hand in boiling water or touch something that's freezing cold, okay? This information is sent back to the parietal lobe of the brain, and then that maps it to the appropriate body part and location. Okay? Proprioception is the sense of position in space. Okay, and these are also mechanoreceptors, and they're found in muscles, tendons, joints, in the inner ear. Okay, and they send information to both the parietal lobe of the brain and to the cerebellum. Okay, the cerebellum needs that information in order to make sure that our intended movements match our actual movements. Okay, if they don't sync up, then the cerebellum will fix that command information and adjust the way that we move. And of course, that information relies on incoming information from our proprioceptors, or our position information, and also our eyes and our inner ear. Okay, and that's how we're able to, appropriately act on our environment in a way that we intend. Okay. The special senses are here, right? Smell is olfaction, taste is gustation, sight, which is vision, right? Hearing, which is audition, and balance, which is actually a type of, you call it equilibrium or proprioception. Okay. So our first one that we're going to look at is olfaction. So we'll take a look here.
The detection of specific airborne molecules, referred to as autogens, results in the sense of smell. As the autogen is inhaled, it moves through the nostril and into the nasal cavity. There it comes in contact with the olfactory epithelium. The olfactory bulb, which lies above the nasal cavity, receives sensory information from the olfactory epithelium. Within the upper nasal cavity, the autogens dissolve in the mucus lining, where they can interact with receptors on cells in a specialized region on the olfactory epithelium.
Okay, so what you're seeing with olfaction is an unusual special sense because it's the only one that doesn't get projected to the brain's router, which is called the thalamus. It gets projected directly back to the temporal lobe of the brain. Chemoreceptors in bipolar olfactory neurons, which project through a region in the base of the skull called the cribriform plate, which is on top of a bone called the ethmoid bone. They dangle in the nasal cavity, and then odorants attach to those bipolar neurons, and when they do, they open ion gates, and then those very short olfactory neurons relay electrical information back to the olfactory bulb, okay, which is right there. And then from the olfactory bulb, we go straight back to the temporal lobe of the brain, and then we draw on memory in order to interpret what it is that we've seen, okay? Some odors can also stimulate pain receptors of the trigeminal nerve, okay, which is cranial nerve 5. An example of an odor that stimulates the trigeminal nerve would be something like, let's say, ammonia fumes, okay? To detect a faint odor, we often sniff deeply, and that's so that the olfactory receptors get adequately saturated with the odorant, okay? Olfactory impulses to different parts of the brain can often trigger memory, and that's because the olfactory bulb and tract are part of a region of the brain called the limbic system, which attaches emotional content to our physical experiences, okay? Without an active limbic system, we would have terrible problems with memory and also with interpreting or expressing emotions to those around us, okay? Our next sense is going to be gustation or taste.
The detection of specific water-soluble molecules found in food or drink results in the sensation of taste. Specialized cells found in taste buds on the tongue contain receptors capable of interacting with molecules found in our food. When a taste receptor is stimulated, an electrical signal is produced by the sensory cell, resulting in an impulse which is transmitted to the brain and results in the perception of taste.
Okay, so what you can see here is the tongue, right? And the tongue has projections on its dorsal surface called papilla, right? Which take on different shapes, right? There are papilla that look like toadstools, and they're called fungiform. There are papilla that look like leaves, and they're called foliate. And there are papilla that look like fibers, and they're called filiform. And then there's valate papilla, which are arranged in a little V shape at the back of the tongue, okay? And they all have the ability to pick up all the basic tastes, okay? Sour, salty, bitter, sweet, and umami are all detectable by all regions of the tongue. It's just that some regions of the tongue are more sensitive to certain tastes than others, okay? But the cranial nerves that send that information back to the brain are 7, 9, and 10, okay? And we'll learn about those when we get to the central nervous system, okay? This information is usually integrated with olfactory information, and that gives us the sense of flavor, okay? So we interpret both smell and taste frequently as one sensation, okay? Okay, next we're going to deal with vision and hearing, okay? And we'll describe the structures that carry these processes out, and their location, and what they do. Okay, so here's a look at vision.
To form images of our environment, light emitted by or reflected from objects must be focused on the retina. Some of the light reflected from the surface of a yield sign is directed toward the eye. Upon reaching the eye, the light is first refracted by the cornea to send it on a path toward the retina. To reach the retina, the light rays must pass through the lens, where a small amount of refraction adjusts the angle of the light to form a focused image on the retina. Central to the sense of vision is the transduction of light energy into electrical energy by cells in the retina called rods and cones. Cylindrical-shaped cells called rods are responsible for the perception of light and dark. Conically shaped cells called cones are sensitive to red, green, or blue light.
So the visual accessory organs include the eyebrows, the eyelids, the conjunctiva, the eyelashes, the lacrimal apparatus, the extrinsic eye muscles, all acting together. Okay, so let's take a look at each one of these components. Okay, the eyebrows are important because they keep sweat out of your eyes, which can be irritating because of the salt content. All right, the eyelids help to protect the eye from drying out and from particulates and physical trauma. Okay, the conjunctiva is a vascular membrane that lines the inside of the eyelid and the white of the eye up to where the iris is, which is the colored part of the eye, and its job is to serve as a source for lubrication of the eye and to protect underlying structures. Okay? And it also provides a blood supply to some of the nearby tissues. The eyelashes are designed to keep particulates out of the eye. The lacrimal apparatus produces tears, which is basically a filtrate of blood, and it washes across the surface of the eye and keeps it moist, and that washes particulates out of the eye, but it also keeps the cornea of the eye, which is the clear skin in front of the iris and the pupil, moist so that it can get its oxygen from the air. Since it has no blood vessels, that's its only way of getting sufficient oxygen. Okay. And then the extrinsic eye muscles, which aren't visible here, are attached to the eyeball, and they move it around. They're skeletal muscles, and we consciously control them, and that lets us look up, down, left, and right, and so on. Okay. The nerve that provides vision is the optic nerve, and it comes out of the back of the eyeball, and it goes straight to the brain's router called the thalamus, and then from there neurons carry that information back to the occipital lobe of the brain, which is the lobe in the very back. Okay, and the muscles that move the eyeball are the oculomotor, the trochlear, and the abducens cranial nerves, which innervate the superior, inferior, medial, and lateral rectus muscles, and the superior and inferior obliques. Okay, so different cranial nerves talking to different muscles, and another cranial nerve providing the visual input to the brain. Okay. The three layers of the eyeball are the sclera, the choroid, and the retina. Okay. The sclera is what's known as the fibrous tunic, and it's the white of the eye. And another component in the fibrous tunic is the cornea, which is transparent. Okay. And the sclera is designed to maintain the shape of the eyeball and to protect the interior structures. And it serves as an attachment point for the muscles that move the eyeball. Okay. The blood supply here is very poor. Okay. And so it has to get its oxygen from moisture. Okay. And also from diffusion of oxygen through what's called the vitreous, and from diffusion of oxygen and nutrients from the choroid, which is the middle vascular layer. Okay. The choroid is the middle layer in the eye, and it has a lot of blood vessels in it. At the front, it forms the ciliary body, which is responsible for making something called aqueous humor, which builds up behind the cornea and in front of the lens, and is also transparent, okay, and keeps those tissues hydrated, but also provides a transparent layer so that the light that passes through it doesn't scatter, okay. The ciliary body also adjusts the shape of the lens so that we can focus on things that are either close up or far away by changing the lens's diameter and width, okay. And it's attached to the lens by special suspensory ligaments, okay. The retina is the neural tunic and contains the photoreceptors. Right? The photoreceptors are the rods that respond to light intensity, and the cones that respond to color. Right? The purpose of the rods and cones is to change light information into electrical information, and then send that electrical information back to the optic nerve, which sends it back to the thalamus, and then the thalamus sends it back to the occipital lobe. Okay? And then the optic disc is just where the optic nerve enters the back of the eyeball. It's also called your blind spot, and it has no photoreceptors. Okay, so it's an area where we don't get any visual information, but fortunately in our visual field, the blind spot in our left and our right eye aren't in the same location, and so one eye compensates for the other. Okay. The rods are generally on the periphery of the retina, and again, they're responsible for changes in light intensity. They get the name rod because their apical segment, the top of the cell, looks like a rod, okay? And it has pigments in there that respond to changes in light intensity by changing the current that flows through the membrane. Okay? The cones get their name from the fact that their apical segments look like a cone, and they have pigments in their apical membrane that respond to particular wavelengths of light. Okay? That provides our color vision. Okay? The cones are concentrated in a region in the retina that's called the macula lutea. Okay? And the highest concentrations of cones in the macula lutea is in a region called the fovea centralis. Okay? So the goal of the eye is to focus the image on the fovea centralis so that you can see effectively. Okay? And so the eye will adjust the thickness of the lens in order to deal with that. Okay? And there are muscles, smooth muscles that do that. Okay? And it'll also adjust the amount of light that comes in by changing the diameter of the pupil, which also aids in our ability to, again, pick up the image effectively. Okay? The posterior cavity is between the lens and the retina and is filled with vitreous humor, which is transparent and also maintains the shape of the eyeball, and also pressure seals the retina against the eyeball so that it doesn't detach. Okay? The only places where the retina is physically attached to the eyeball are at the optic disc and at the anterior border of the retina, which is called the ora serratia, which means serrated border. And that's why if you take a shot to the head, it's pretty powerful. You can knock your retina loose and go blind. Okay? The anterior cavity is between the lens and the cornea and is filled with aqueous humor, which is made by the ciliary body from blood, and that flows into something called the canal of Schlemm, which we'll see here in this diagram, and then that drains, eventually gets back into the blood supply. Okay, sometimes when the canal of Schlemm is blocked or the production of the aqueous humor is too high, you can increase the pressure in the eyeball, and that can lead to crushing the retina, and that could cause blindness, and that's a condition you've probably heard of known as glaucoma. Okay. Muscles that move the eyeball include the extrinsic muscles that move the eyeball in the orbit, and the intrinsic muscles that adjust the diameter of the pupil, which are around the iris, and the ciliary muscles, which adjust the shape of the lens. Okay, so these the muscles in the pupil and the muscles around the lens are smooth muscles. Okay, and they're innervated by cranial nerves, and the muscles that move the eyeball are skeletal muscles and are innervated by a different set of cranial nerves. Okay, so you can see the muscles that move the eyeball here, right? There's the superior and inferior rectus. Okay, there's superior, there's inferior down there. There is the lateral rectus, and the medial rectus is on the side of the eyeball we can't see. And then there's the superior oblique and the inferior oblique. Okay, the lateral rectus is innervated by the abducens nerve. Okay, and the superior oblique is innervated by the trochlear nerve, and all the rest are innervated by the oculomotor nerve, which is cranial nerve 3. Okay. The intrinsic eye muscles change the diameter of the pupil or the shape of the lens. Okay. The iris contains circular muscle that is going to respond to impulses, right, to either increase or decrease the diameter of the pupil. Okay, and then there are radial muscles that do the same. Okay, contraction of the circular muscles is going to constrict the pupil, and that's a condition known as miosis. Constriction of the radial muscles causes dilation of the pupil, and that results in mydriasis. Okay. The receptors in the circular muscle are muscarinic receptors, okay? And in the radial muscles, they're alpha 1s. Okay? The ciliary muscles are going to respond to changes in light intensity, okay? By either, they're going to respond to changes in the distance that the object is from the eye by either causing the lens to fatten or to become thinner. Okay, the closer the object is you're looking at, the fatter and thicker the lens will become. Okay, and the further away it is, the narrower the lens will become. All right. Pupillary function is evaluated by noting the size, the shape, and the reactivity to light. The acronym PERLA, P-E-R-R-L-A, is an assessment term for pupillary function, which means pupils equal, round, reactive to light, and accommodating. Okay, if you assess it as pupils fixed and dilated, that can be an indication of death. Okay. Some eye disorders. Okay. Ptosis is a drooping eyelid. A hordelum or a stye is basically a blockage of ocular secretions that results in inflammation. Pink eye is an inflammation of the conjunctiva. Okay, it can be viral or bacterial, or it can be the result of toxin exposure. And strabismus is essentially the eye not tracking with the normal direction of vision. Okay, and nystagmus is the eyes tracking either back and forth or round and round, and that can be an indication of vertigo. The pupil constricts when it's exposed to light. The other pupil will also constrict, and that's what's called a consensual reflex. And these muscles come, again, from the radial and the circular muscles that surround the pupil. Okay, and the ciliary muscles come from the ciliary body, and they change the lens. Okay, So one of the things that happens when an object gets closer to the eye is a series of events called accommodation, right? And what accommodation is where the lenses get thicker, okay? The pupils constrict, okay? And the eyes converge, right? And that makes it easier for you to see an object that's close up, okay? So some of that stuff is involuntary responses, and some of that stuff is the result of voluntary responses, but they're all part of cranial nerve activity. Okay? Refraction is just the bending of light. Okay, light has to go through 4 transparent layers before it gets back to the macula lutea, and then is perceived as a sharp image. Okay, remember that the macula lutea, in particular the fovea centralis in the middle of the lutea, is where the bulk of the cones are, and that's where our greatest visual acuity is. Okay, so the transparent layers are the cornea, and behind that the aqueous humor, behind that the lens, and behind that the vitreous humor, right? And they all bend light so that we hit the macula totally in focus. Okay. So we've talked about accommodation, but emetropia refers to the ability of the eye to refract light without corrective lenses, which means that you have good vision. We'll call that 20/20, right? Refraction is just the bending of light, and we have to do that in order to get the rays to focus on the fovea, okay? If we don't do that, the image is going to be blurry. So what will happen is that in individuals that have what are called errors of refraction, the image doesn't get to the fovea in focus. Okay, examples include myopia, in which either the optical components of the eye or the dimensions of the eye are such that the image comes into focus before it gets to the fovea, and the image is blurry. Okay, and we generally fix that with concave lenses in your glasses, or with laser corrective surgery to flatten out the cornea, or we're able to do that with contact lenses that do the same thing, right? Hyperopia is where the image comes into focus past the fovea, right? And so what we have to do there is use glasses with convex lenses, or we have to use laser corrective surgery to thicken the cornea, or contact lenses to do the same thing. And then in astigmatism, you either have an irregular surface in the lens or in the cornea, and the result of that is that some of the image comes in too soon and some of the image comes in too late, and you get a blurry image. And the fix for that is either bifocals, okay, in your glasses, or you can have laser corrective surgery to smooth the cornea, right? Or sometimes, if it's bad enough, we can replace the lens, if it's the lens that's the problem with the astigmatism. Okay, we have night vision. In low light conditions, note that the pupil dilates and lets more light into the eye, and the light rays scatter along the periphery of the retina and stimulate the rods, and that gives us black and white vision, basically. In well-lit environments, the pupil is constricted, and it directs the light towards the macula lutea, okay? And the image is produced by the stimulation of the cones and is nice and sharp, all right? So that, again, is sort of talking about the difference between the ways the receptors are localized, right, and the light conditions that provide them. The visual pathway shown here is that light bounces off an object, and then first it passes through the cornea and the aqueous, and then the lens, and then the vitreous in the eyeball, and then it's going to hit the macula lutea. Okay, then the retina is going to relay that information back to the optic nerve, which is going to relay it back to the thalamus. Okay, and you can see that part of the pathway here. Okay. That's the part where we hit the thalamus. And then the thalamus is going to send that information back to the occipital lobe of the brain. Okay? It's interesting that the right side of the brain deals with one visual field, and the left side of the brain deals with the other. Okay, um, but both visual fields are represented in both eyes. All right, so you can see kind of how they project. The nasal fibers of the optic nerve cross at a region called the optic chiasm, and the temporal fibers do not cross. Okay, so they stay on the same side and project back, right? So this left side of the brain, left occipital lobe, is going to be dealing, if you trace it, with the right side of the visual field in both eyes, and vice versa. Okay? That's the pathway, right? We talked about it. Okay? We talked about myopia and hyperopia as well, and astigmatism, and how that's caused. Okay? And so our next topic is hearing. So let's take a look at the ear.
Sound waves strike the tympanic membrane and cause it to vibrate. Vibration of the tympanic membrane causes the three bones of the middle ear to vibrate. The footplate of the stapes vibrates in the oval window. Vibration of the footplate causes the perilymph in the scala vestibuli to vibrate, which in turn causes displacement of the basilar membrane. Short wavelengths from high-pitched sounds cause displacement of the basilar membrane near the oval window. This movement is detected by hair cells of the spiral organ, which are not visible in the animation. Long wavelengths from low-pitched sounds cause displacement of the basilar membrane far from the oval window. Again, this movement is detected by hair cells of the spiral organ, which are not visible in the animation. When the vibrations reach the perilymph in the scala tympani, they travel to the round window where they are dampened.
Sound waves are gathered by the pinna and directed down the auditory canal to the eardrum or tympanic membrane. The mechanical energy of the sound causes the eardrum to vibrate. This in turn causes vibration of the three middle ear bones, first the malleus, then the incus, and finally the stapes. These tiny bones amplify the vibrations. The stapes comes in contact with the inner ear at the oval window. The fluid-filled inner ear is a coiled structure called the cochlea. Vibrations conveyed to the fluid cause bending of hair cells. If we examine this cross-section of the cochlea, the tectorial membrane can be seen at the top. In it are embedded the tips of hair cells, which are shown circled in yellow. When the hair cells bend in response to vibration, the rate of firing by the neurons at their bases changes. These signals are conveyed to the brain and perceived as sound.
Okay, so as you saw in the previous two videos, the job of the ear primarily is to turn sound, which is a pressure wave going through gas, air, into electrical information that's going to get back to the brain through the eighth cranial nerve, which is the vestibulocochlear nerve. Okay, the pinna is like a funnel, and it focuses the sound waves until it goes through the external auditory canal to the tympanic membrane or eardrum. The tympanic membrane shakes, and that causes these bones to shake. This is the malleus, and this is the incus, and this is the stapes, and they transfer the vibrations to the oval window on the surface of the vestibule. But what that does is that causes a liquid inside the cochlea, this little snail-shaped organ, to move, and that liquid is called perilymph. Okay, and when the perilymph vibrates, that causes another chamber, the cochlear duct, which is filled with endolymph, to vibrate. And then there are hair cells that are on the surface of the basilar membrane in the base of the cochlear duct which bounce up and down as that fluid shakes, and the hairs on the top of these hair cells, which are actually stereocilia, they're not really hairs, hit something called the tectoral membrane, and that opens ion gates, and that lets current flow into them, and then that's going to be perceived by the brain as sound. So the eighth cranial nerve will project back to the thalamus, and then that'll project to the temporal lobe of the brain, and that'll provide our sense of hearing. Okay, so that's basically the pathway that sound takes. Okay, the three parts of the ear, the external, middle, and inner ear, have different functions, right? The external ear funnels the sound. The middle ear is going to turn the sound into a vibration that the inner ear can turn into electrical impulses. Okay, this is all embedded in the temporal bone of the brain, or the skull. Okay. The other material out here we'll talk about in a bit is the balance apparatus. That's the vestibule and the semicircular canals, and they provide our ability to stand up straight when we're moving or when we're standing still. Okay. The vestibule responds primarily to gravity, and the semicircular canals respond to rotational movement. Okay, so as you spin, fluid in these moves, and that causes the bending of stereocilia, and that's perceived as balance information. In the vestibule, gravity is doing that. So as you tilt your head, that bends stereocilia, and they produce information about balance when you're not moving. Okay, so we'll get into more detail about the balance stuff in a bit. The external ear, made up of the auricle, which is the funnel-shaped portion, it's also called the pinna, and the external auditory canal. And the middle ear runs from the tympanic membrane all the way to the lateral border of the vestibule. Okay, and it contains the Eustachian Tube, which runs from the middle ear to the nasopharynx, right, the part of your throat that's behind your nose. And the purpose of that tube is to regulate the pressure on either side of the eardrum so that it can vibrate. Okay, and this is one of the ways that you get ear infections, is that you get bacteria and infected mucus working its way up through that tube, and that produces gas and swelling behind the drum, and that generates pain, and then the drum can rupture, and that causes the drum to scar, and that causes the drum to be less responsive to sound. Okay? The inner ear is the cochlea, the vestibule, the semicircular canals, and also the connection of the eighth cranial nerve, which is the vestibulocochlear, right? The Eustachian tube, again, is that pressure regulating tube, okay? In young children, it's usually short and pretty horizontal, and that's why it's easier to get ear infections as a child. In adults, it gets longer and more vertical, and it gets more twisty, and the result of that is that it will be less likely to generate an infection. Okay, so that's one of the reasons why we kind of grow out of middle ear infections. Okay, longer and more vertical. Okay, the organ of Corti is that part of the cochlea that I was discussing earlier, which is in the core of the cochlea. Right, this is where the endolymph is, and the perilymph would be here and here. This stuff. The liquid here shakes and shakes, and the liquid in here shakes, and then these guys bounce up and down, and that right there is the tectoral membrane. This is the basilar membrane, and then when these little hairs hit this flap, they open up the ion gates, and then the current flows in, and then that's what we perceive ultimately as sound. Okay, and again, this is all embedded in the temporal lobe of the brain. The cochlear duct and the rest of the apparatus is filled with endolymph, and it's only in the cochlea that we have also perilymph. Okay, so we have really three sets of tubes, right? The scala tympani, the scala vestibuli, and the cochlear duct, right? So endolymph here, perilymph here, and here. Okay. So that's the way that the hearing occurs. Okay, sound waves, tympanic membrane. Ossicles hit the oval window, organ of Corti responds. We send impulses back to the eighth cranial nerve, and that goes to the thalamus, and that goes to the temporal lobe of the brain, and that then is going to be drawing on information from the the auditory association area, which is also in the temporal lobe, and then we can connect that sound to language or noise that we've heard in the past. The organ of Corti is really the contents of the cochlear duct, including the endolymph and the hair cells and the tectoral membrane and the basilar membrane. Okay. So when hearing doesn't occur, we have blockage. Right? We can have problems in getting the sound wave back to the cochlea, or we can have problems getting the electrical information back to the brain. Okay, so you can have damage to the vestibulocochlear nerve, and we can have damage to the cochlea, and that's called sensorineural deafness. Okay, so anything from the cochlea back is going to interfere with the signal getting to the temporal lobe of the brain. Anything but before the cochlea, like damage to the ossicles or damage to the drum or damage to the external auditory canal, is what we call conductive deafness, and that is basically blocking the sound wave from ultimately impacting the cochlea, and that can be usually fixed surgically. Damage to the cochlea or the cochlear nerve can be fixed through what's called a cochlear implant. Okay, so what we can do is basically screw a microphone into the mastoid process of the skull, which is the bump right behind your ear. Okay, and then we run wires or electrical leads into the cochlear nerve, and then the cochlear nerve, of course, goes back to the temporal lobe of the brain, and then we can stimulate that part of the brain, and then the brain has to learn to associate that stimulus with sound. Okay, so it usually works better in people who have had hearing and lost it than in people who have never heard before. All right, so that's a critical point. Okay, the next thing we want to do is look at our balance apparatus.
The ability to sense head position is achieved by monitoring sensory organs in the saccule and utricle of the inner ear, called maculae. The hair cells in the macula are key to the vestibular sense. Each hair cell has a large cilium referred to as the kinocilium, and many smaller cilia called stereocilium. When the cilia is bent towards the kinocilium, the firing rate of the hair cell decreases. Within the macula, the cells are embedded in a gelatinous layer that is weighted by otoliths.
So, as we discussed before, right, changes in head position cause fluid within the balance apparatus to move. This bends the stereocilia and it triggers nerve impulses that travel along the vestibular branch of the 8th cranial nerve to different parts of the brain, including the cerebellum, the midbrain, and the temporal lobe. And after receiving this information, the brain sends motor information to the muscles, and that helps you maintain your sense of balance. A person with an inner ear disorder is going to suffer from fall-related injuries because they're going to lose their ability to balance, right? We should point out that the vestibule here is where the static equilibrium comes in. Okay, and this is what responds to gravity or head tilt, whereas the semicircular canals respond to rotational movement or spinning, because the hair cells are located in these little swellings at the base of the semicircular canals that are called ampullae, right? And remember that endolymph fills all of this, right? So there's always fluid in here, right? Okay. Again, any questions? Make sure and contact me, and I will see everybody in the next podcast. Thank you very much.